Welcome back to BioB 160 Principles of Living Systems. I'm Ruth Reitzman, your instructor for the course, and this is part 1 of cell structure. In this episode, we'll take a tour of the cell and learn about the basic unit of all living organisms. Let's get started. We often say that cells are the basic unit of life. So what are the characteristics of life, and why are cells considered to be the simplest unit of life? As we discussed at the very beginning of this course, the characteristics of life include reproduction, response to the environment, growth and development, energy processing, regulation, evolutionary adaptation, and order. All of these are evident in cells, which are the smallest living unit that has all of the characteristics of life. In fact, there are more single-celled organisms on this planet than anything else, and they are certainly very alive. As we study cell structure, it's also helpful to keep in mind their size relative to things that we're familiar with in everyday life. Most cells are too small to see with our naked eye, and we need microscopes to study them in detail. Both the light microscope and the electron microscope are important tools for cell biologists to see and understand cell function. One of my favorite websites is the University of Utah's Learn Genetics site, www.learngenetics.utah.edu. One of their features is the Cell Size and Scale Interactive Viewer. Let's take a look at this site. As we view this site, we can see that this is an interactive scale, and up at the upper left-hand corner, you can see that we have an indication of what size we're in. So we're in the 1 millimeter range at this point, and we can see things that we would see with our naked eye slightly enlarged at this point: the coffee bean, the grain of rice, and regular type font. If we scroll in, we can start to see other things. The sesame seed and the grain of salt has expanded a little bit. And now we're scrolling into simple protozoa like amoeba and paramecium. And if you notice, the scale here has changed. So now we're on the micrometer scale. If we go a little farther, now we can see skin cells and red blood cells. And if we get a little closer, we can start to see prokaryotic cells, which we'll talk about in just a second. And now we're on the micrometer scale. So at this point, we've almost exceeded what we can see with the light microscope, and we're moving into what we need the electron microscope to see. If we go a little closer, now we can start to see the subcellular particles like the mitochondria and lysosome. These organelles we'll talk about in a second. And now we can start to see virus sizes. So measles virus, influenza, HIV, and here we have rhinovirus, which is the cause of the common cold. And notice now we're on the nanometer scale, which is definitely in the electron microscope range. And if we go a little closer, we can see the ribosome, antibody molecules, and things we talked about with the macromolecule section. So RNAs, phospholipids, the amino acid methionine, and finally we get down to the water molecule and the carbon atom. So this is a scale we'd need X-ray crystallography to take a view at. So if we go backwards, we can see that we're zooming out again. And of course, no one microscope is going to let us see all of these things, but with a combination of techniques and this composite amazing animation, we can see a lot of information. So if you want to see this again, here's the URL, learn.genetics.utah.edu. Very fun site to look at. Now that we know something about the size and scale of cells, let's talk about different types of cells. There are two different types of cells in the world: prokaryotic cells and eukaryotic cells. Prokaryotic cells include the bacteria and the archaea, and while organisms such as protists, fungi, animals, and plants are made of eukaryotic cells. The main difference between the two types of cells is the presence or absence of a nucleus, the organelle that contains DNA. Both prokaryotes and eukaryotes have DNA, it's just that prokaryotes have naked DNA in their cytoplasm. In fact, prokaryotic cells lack any membrane-bound organelles, while eukaryotic cells have many different organelles, which we'll be discussing in this episode. And in size, prokaryotes are much smaller than eukaryotes. Prokaryotes are usually 1 to 10 micrometers in length, while eukaryotic cells are 10 to 100 micrometers. This chart summarizes these differences, so be sure to review this before the exam. This diagram shows a typical prokaryotic cell, and you can see that it's pretty simple. It has a cell membrane, a cell wall, and just a few internal structures, but no internal membranes. However, this is a very successful group of organisms, and they can be found pretty much anywhere on the planet, from polar ice caps to hydrothermal vents, deep in the ocean, and to in and on the human body. This slide is a photograph of eukaryotic cells. Actually, they are epithelial cells that are found on the inside of our mouth, with bacteria clinging to them. This is a good view of the size difference of eukaryotic versus prokaryotic cells. And don't worry about having bacteria in your mouth. That's perfectly normal. As we tour the eukaryotic cell and explore its structure and function, we'll try to answer the questions: what features are found in all cells? And we'll also examine the features that are unique to eukaryotic cells. This diagram from your textbook shows a composite drawing of a eukaryotic animal cell. This drawing will be helpful to refer to as we discuss the various structures in the animal cell. This diagram shows a eukaryotic plant cell. You should also refer to this diagram and be able to compare it with the structure of the animal cell. We'll begin by looking at the plasma membrane. All cells have plasma membranes, also called the cell membrane. The key feature of the plasma membrane is its selective permeability. In other words, it allows some, but not all, substances to pass through, so it regulates what comes in and goes out of the cell. This is a very important function because the plasma membrane divides the inside from the outside of the cell, so nutrients must be brought into the cell and waste products must be released. The plasma membrane is composed of a bilayer of phospholipid molecules with proteins embedded or floating in the membrane. In our next episode, we'll talk more about membrane structure and the ways that substances cross the membrane. Another important structure in the eukaryotic cell is the nucleus. The nucleus can be thought of as the information center of the cell. It contains the cell's genetic information, which is specified in the cell's DNA. Eukaryotic DNA is combined with proteins to form chromosomes. When chromosomes are tightly packed together, they form chromatin. Another structure in the nucleus is the nucleolus, the area of the nucleus where ribosomes are formed. The nucleus is surrounded by a double membrane. This diagram shows the nucleus with its double membrane and its connections to the cell's endomembrane system. The area of the endomembrane system closest to the nucleus is the rough endoplasmic reticulum, or rough ER. This diagram shows the nuclear pores, which allow the exit of ribosomes from the nucleus. Ribosomes are the protein factories of the cell. They are sometimes called the workbench for protein synthesis. Ribosomes consist of ribosomal RNA plus ribosomal proteins. The ribosomes in eukaryotic cells are found in two locations, free in the cytoplasm or bound to the rough ER. Also, we should mention that the ribosome consists of two parts, the large subunit and the small subunit. We'll learn more about ribosomes and the process of protein synthesis, also called translation, later this semester. This diagram shows the rough endoplasmic reticulum with the ribosomes bound to it. Our next slide will examine the endomembrane system of the cell. This system is responsible for the production and transport of proteins, and it consists of the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and finally the plasma membrane. First, we'll look at the endoplasmic reticulum. This segment of the endomembrane system has two parts, with two very different functions. The rough ER has ribosomes on its surface and is involved in protein synthesis. The smooth ER lacks ribosomes and is responsible for lipid synthesis. This diagram is a photomicrograph of the endoplasmic reticulum. The diagram in the photo, taken with the electron microscope, highlights the difference between the rough and smooth ER. The rough ER looks like flattened sacs and has knobby bumps of the ribosomes that are attached to it. The smooth ER looks like tubules and lacks the knobs or bumps on its surface. The rough ER is involved in protein synthesis. Any protein that is destined to be contained in a membrane-bound vesicle will be synthesized within the rough ER. The rough ER then distributes the proteins in transport vesicles, which travel to the Golgi apparatus. In addition, the enzymes for membrane synthesis are located in the rough ER. The smooth ER's functions are very different from the rough ER. The smooth ER contains enzymes for lipid synthesis. In some cells, the smooth ER has enzymes to detoxify poisons and drugs. And in muscle cells, the smooth ER stores calcium and releases it when the muscle cell is signaled to contract. The next region of the endomembrane system is the Golgi apparatus. In the electron microscope, it looks like a series of flattened sacs called cisternae. The Golgi apparatus can be thought of as the shipping and receiving center of the cell, a sort of UPS or FedEx distribution center. In this case, the Golgi receives packages or vesicles containing proteins made in the rough ER. The Golgi then modifies these proteins and sorts and packages the proteins for distribution to transport vesicles. This figure shows the Golgi apparatus. The cis face is the side closest to the rough ER and is the receiving side of the Golgi. The trans face is the side closest to the plasma membrane and is the shipping side of the Golgi apparatus. Another important component of the endomembrane system is the lysosome. The lysosome is the digestive compartment of the cell, and it is a membrane-bound sac filled with hydrolytic enzymes that carry out digestive or hydrolysis reactions on a variety of macromolecules. You'll recall from our discussion of macromolecules that a hydrolysis reaction is the reverse of a condensation reaction. So hydrolytic enzymes are required to break down large macromolecules into their monomer units. The lysosome carries out two types of digestive functions, phagocytosis and autophagy. The lysosome uses phagocytosis when it engulfs food particles. In phagocytosis, the food vacuole is fused with the lysosome, forming a digestive vacuole. Then the lysosome's hydrolytic enzymes are activated and the food molecules are digested. This process also occurs when our white blood cells engulf potentially harmful bacteria and then kill them and digest the bacteria. Great way to get rid of a potential problem. Another type of digestive function is autophagy, or literally self-eating. In autophagy, the worn-out cell parts are engulfed within a membrane vesicle and then fused with the lysosome. As we saw in phagocytosis, the lysosomal enzymes become active and the molecules contained in the vesicle are digested. It's sort of like cellular cleanup. We get rid of old, worn-out structures and then make new ones. Interestingly, a recent study found that exercise induces autophagy and helps keep cells young. In fact, the investigator who conducted the study was so impressed with the results, she actually went out and bought a treadmill and started exercising herself. Just another reason why going to the gym or heading out for a run is good for you. Vacuoles are a very important and diverse part of the endomembrane system. Vacuoles perform a variety of maintenance functions, including storing food in food vacuoles, removing water in contractile vacuoles, and storing water and other materials in the central vacuole of a plant cell. This diagram shows a plant cell's central vacuole, which is a very large structure in the center of the plant cell. The central vacuole contains water and provides turgor pressure and strength to the plant cell. And it will decrease in size if the cell becomes dehydrated. The next time you notice that your houseplants have wilted, be sure to give them a good watering so their central vacuoles can fill up again. The next few slides show the relationship of the different organelles of the endomembrane system. First, we see the nucleus and the nuclear membrane with its connection to the rough ER. Also, we can see the smooth ER. Next, we see the Golgi apparatus with its cis receiving face and trans shipping face. Finally, we can see a transport vesicle from the Golgi merging with the plasma membrane to release its package of proteins. This concludes part 1 of cell structure. In the second part of this episode, we'll explore the mitochondria, chloroplast, and the cell cytoskeleton. Thanks, and have a great day.